Thanks. I feel a bit of an intruder here because first I'm going to present some of the applied stuff, and secondly, I'm not employed within the Maria de Maeztu funding. So my funding actually comes from the Spanish Ministry of Science. So this is the project from which I'm working, which is related to trying to analyze how noise and heterogeneity influences cancer. But today I'm not going to talk about this because I joined the project recently and I don't think it's ready to be presented yet. So I will tell you some small story which we have been doing in the past years, a little bit just to illustrate what type of things we are interested in mass biology, so you can get an idea. And one of the things that we usually find particularly interesting is how cells behave. So we try to understand this. And we're also interested in the emergent properties which result from this individual cell behavior, so that you can see then rather on the collective level when you put a bunch of cells together. So, and in this regard, one of the topic which is rather popular in mathematical biology is to study pattern formation, what we call. So if you put a bunch of cells, for example, a cell monolayer, you can see that through the communication between those cells, you can see various patterns occurring. So for example, you can already see on this small image here that there is a pattern going on. And this is so in this talk, I will be speaking about a particular case of the cell-cell communication, which is done through what we call notch signaling. And this is the work that we've been doing together with Tomás, who is the PI here at the Computational Mathematical Biology Group, and also our collaborators from Oxford, Helen Byrne and Philip Maini. So I can just start perhaps by giving you a couple of examples where you can find notch signaling. So it is an evolutionary conserved pathway, which is really present in many cells in different organisms. So one of the classical examples that we usually consider is the bristle patterning in the fly snotum. So you can see that the positions for the bristles to grow would be defined by different gene expression. So you will see a very different pattern, but also due to notch signaling, also defines the positions of the veins in the fly wings. So you can see that in this case, this would be the stripe pattern. We will not cover this, but let me show you the examples that I will talk about. So one of them is the cellular patterning that can be observed in intestinal crypts. So one of the, if you do not know, so one of the things, I don't know how to use this. So one of the things is that the tissue of the intestine is covered in millions and millions of small invaginations. And the very bottom part of those invaginations, you can see it here, this tiny thing, is called intestinal crypt. So we can see it better in this cross-sectional image. And we will be particularly interested actually in the bottom part of those crypts, which is called crypt niche. So there is some cell patterning going on in here, and this is also notch dependent. And another biological system that we also studied is the growth of new blood vessels. In this case, the blood vessels, the inner layer of them is composed by cells which are called endothelial cells. And when they perceive some stimulation for the new blood vessel to grow, so what they do, they start breaking apart this basement layer that they have around, and they start sprouting towards the stimulate that they perceive. And there is also some cell patterning going on in here, so you can see them. So I will tell you about those particular cases. And how we ended up studying those subcellular models, how the cell signaling going on, is because we wanted to actually do some agent-based simulations for this type of the biological systems. And of course, if we didn't want to prescribe cellular behavior, and we rather wanted it to get it like through the real signaling, depending on what cells perceive from the environment, we needed to study first what is the cellular machinery that is going on that gives you this type of patterning. That's how we ended up here. So I would like to describe you first what we call the classical pattern that you get using notch signaling. It's a type of the lateral inhibition mechanism. I'll explain you what it means. So imagine you have a cell 1 and a cell 2 in this case. And there is a receptor and a ligand. So if you don't know what those are, receptor is like a keyhole, and the ligand is like a specific key that can open it, right? So they can bind together. So in this case, if you have two neighboring cells, then the delta ligands can bind, well, we call it transbinding because they belong to a different cell. can bind to the notch receptor on the cell 1, and this initiates notch signaling in this target cell. And then when those downstream genes start expressing, what happens is that they inhibit production of the delta ligand itself. And then the same behavior can be mirrored to the other cell. So if we do the modeling and we write down the corresponding chemical reaction, we do the analysis and we try to implement those equations on a lattice, let's say, of interacting cells, this is type of the pattern that you get. We call it the classical salt and pepper, because in this case, different cell phenotypes, they would be separated by just one cell of the other phenotype. And I'll try to use the same color scheme. So in this case, the red color would be always cells which express high notch, and green cells would be expressing low notch. And the delta, because we have this relation here, right? So we know that the delta ligand is essentially reversed, just for you to know. So we call this the lateral inhibition mechanism, because essentially the cell, in this case, this is the low notch. So this central cell would inhibit all its neighbors to acquire the same phenotype. So you laterally inhibit your neighbors to become the same as you are. So this is the classical pattern. So usually I will omit all details which are related to the cell 2, because in reality there's not just one neighbor. Now you can have a number of neighbors depending on the cell geometry, and I'll just be calling them neighbors, and I will assume that there is some amount of the external ligand and receptor that the target cell can perceive. So we have something like this. And then why do we actually need to go beyond this classical pattern? We can take a look at what goes on in the different biological systems that I presented to you before. Firstly, interactions in endothelial cells, they are characterized by longer range connections. So we can take a look at this experimental image. In this case, this is the small sprout, so a blood vessel growing. And this red and green color, those are actually the same markers. Well, they express the same thing. So you can see that those, this is the same phenotype, and then they're separated by cells of other phenotypes, because the white color indicates cell nuclei. So I'm just gonna do this for you to see the pattern better, because otherwise it's difficult. So we can see that there are two cells which are low notch, and they're separated by several cells which are high notch. So in this case, several of them. So this is a different pattern. We call it a long range interaction. And another interesting case would be the inversion of this classical pattern for the creep niche. In this case, the high notch cell is the stem cell, which is this tiny thing in here. It's squeezed between other cells which are very roundish and bulky. We call them Paneth cells, or the equivalent. And in this case, well, you can see it better if you look. So this is the creep niche in the cross section, but this is if you look at it from below. So in this case, the stem cells would be those little guys in here, and this big round guys would be the Paneth cells. So if you use the same color scheme to see it better, this is what we have. And we can notice that the high notch cells in this case are surrounded by the low notch cells. So this is the inversion of the pattern. So, and again, since we wanted to study those systems and we wanted to do the simulations of the blood vessel growth, or to understand what happens in the creep niche, we started doing subcellular signaling for those cases. And all of them are given by the notch signaling. So I'm going to start by talking to you like what is going on in endothelial cells, and then we'll talk about the crypt. So this is just a simple cartoon to illustrate you the notch signaling. So in this case, we have the delta ligand, which belongs to some neighboring cell, can bind to the notch receptor, and then the notch signaling is initiated in the cell nucleus. So in case of endothelial cells, this pathway, notch pathway, is coupled with a second pathway, which is called, which is coming from this VEGF, which stands for vascular endothelial growth factor. So this guy is in here, it's floating in the external space. It can bind to its to its receptor here. And then when this happens, it induces the upregulation of the production of delta ligand. And then the coupling between those two pathways is also in this connection in here, so that notch signaling actually down-regulates the production of this receptor for VEGF. And then when this happens, this link is weakened. There are more things going on in general, which I will not talk to you about, but some things, for example, that notch signaling is autocatalytic. So when it's going on, it induces the production of the notch receptor. And there is also mutual inhibition between the ligand and receptor when they find themselves on a cellular membrane. But anyway, so if you write down equations corresponding to this chemical system and you analyze them, so you can find something like this type of plots that we usually do just to see how many steady states we have and what is the behavior of the system, depending on the signals that the cell perceives. So in this case, what I will be plotting here is the amount of the external stimulus, VEGF, which is perceived by the cell, and the amount of the ligand from the neighboring cells. And we can see, so here I plot those two regions are the monostability regions. So one of them, the red color, corresponds to the high notch, as we had before, and the other one corresponds to the low notch. So there is only one steady state available there. And the interesting thing is going on in between, where we have the biostability regime. So this is actually where the pattern can occur, similar to what we saw before. And what I just would like to explain to you is why this biostability region opens up like this with this curvature for increased values of the VEGF. And perhaps we can see it a little bit better if we take a look at the bifurcation diagrams. So I'm going to be plotting here those two bifurcations. They're very simple. They're just saddle node bifurcations for all the cases, for the notch and for the delta in the target cell. First thing just to notice that the expression is reversed, as I was mentioning already before. So when the notch is low, delta is high, and the other way around. And so the biostability is between those saddle node bifurcations in here. So what happens for the low notch conditions is that the amount of delta which is perceived by this target cell in the middle is rather low, because we have this connection, right? So when the VEGF is low, this link is not working. But then when we upregulate the VEGF, then the amount of delta, which is perceived from the neighbors, of course, increases. And then it means that if you want to maintain the same pattern to happen, this biostability region needs to shift and open towards the right-hand side. So if I make this plot animate for the increased VEGF, you can see how they're moving and they're opening up. So this is just to say, this is only to maintain exactly the same pattern, just for the variety of different VEGF levels. I haven't told you anything about the long-range interactions, so we can, let me just mention this, how we actually get there. So one thing that I didn't also mention you about endothelial cells is that they don't have these round shapes. They're usually characterized by elongated shapes. And they also interact a lot with different membrane protrusions, which are in reality much longer than I'm picturing here. They can be up to 3 cell diameters. So one trick to do, if you want to model the patterning, is to forget about cell shape and just to think that we are going to model patterns between the cell nuclei. And in this case, the interaction can be actually non-local. So, well, non-local, I mean, because it's beyond the nearest neighbors. So we can define this radius of the interaction. And if you vary this, we can find different results going on. So for the radius of interaction, which just corresponds to the nearest neighbors, we can recover what we call the classical pattern, because there is one cell separation between different phenotypes. And then if we increase it, of course, the pattern gets sparser. So in this case, this is how we recover the pattern that we saw in the experimental image that I showed you in the beginning. So in this case, you can see the low notch cells, they're separated by several cells of the other phenotype. So this is one of the models that we obtained when we started using, when we started modeling the growth of blood vessels, and we used it. And now the second case about intestinal crypts, which I find a little bit more interesting, in my opinion. So in this case, again, we have the notch signaling. This is what we had before. We can give it a little bit more detail because it is important here. So one thing is when the notch receptor binds to this delta ligand on the neighboring cell, a part of this receptor is cleaved. This part is called notch intracellular domain, which is here. This notch intracellular domain is translocated to the cell nucleus, and there it initiates expression of different downstream genes. One of them is called Hes, and then there is this feedback going on. So another player in here is beta-catenin, which is always present in the cytoplasm of the cell, and it can interact with the notch pathway, firstly by forming a complex with notch intracellular domain, or by directly upregulating the expression of Hes in here. So in case of crypt, the notch signaling is coupled to another signaling pathway, which we called Wnt. This is also an external activating stimulus. Let me tell you how it is coupled. So usually the amount of beta-catenin in the cytoplasm is controlled because there is a special complex of proteins, which we call destruction complex. It binds to beta-catenin and then it induces decay of it. So usually you have normal controlled levels of beta-catenin within the cell. However, when we get Wnt being present in the extracellular space, this Wnt prevents the formation of destruction complex, and then of course the beta-catenin doesn't decay, its levels rise, and then it intervenes with the notch signaling which happens in here. So to mention you that the Wnt gradient which cells perceive along the crypt is rather non-uniform. So in this case, green color shows the gradient of Wnt. So this is the upper part of the crypt. You can see there is little green because here its levels are rather low, and it's really high in the bottom. So what we call crypt niche. So if you want to make a diagram of this is going to be something like this: really high in the bottom and rather low. And the cell phenotypes which are known to be along the crypt. So in the upper part, we have the classical lateral inhibition pattern in this case, and the phenotypes would be absorptive for the high notch cells and secretory for the low notch cell in the middle. But in the crypt niche, as I already told you, there is something going on and the pattern becomes inverse. And we have a stem cell, which is a high notch, which is in the center. And we have Punnett cells, or their equivalent for the small intestine, which is called deep crypt secretory cells, which are low notch. And this is given by the same notch signaling. Okay, so when we started working on the crypts and tried to do the modeling, we ended up discovering here. So we discovered that there are those cells there, and we wanted to model this. So we wanted to make sure that we capture all different phenotypes for the corresponding places in the crypt. So and we actually did this. So if you write down the equations corresponding to this chemical system and you do this correctly, so you can find this type of plot. In this case, so again, I'm going to be plotting here the external stimulus Wnt, and here I will be plotting the amount of ligand which is sensed from the neighboring cell as before. And the colors as before, so we have the monostability region where there is only one cell phenotype that can happen, which corresponds to the high notch signaling, another one for the low notch, and this is the biostability region where we actually can get the patterns. So if we analyze this, what we can notice is that in this case, for the low Wnt signaling here, the pattern is only allowed that when the amount of delta is low. And this is exactly the classical inhibition pattern that we have in here. Because in this case, the amount of delta is going to be, when the notch is high, as I told you before, the delta is low. So this is this pattern. And then as we go along, you see that this pattern moves. And in this case, for high Wnt, what we obtain is that this pattern is allowed for high amounts of delta. So in this case, the pattern becomes inverted because low notch cells, they have the high delta. So in reality, if you go along for the increased Wnt concentration, you actually get the whole transition from one phenotype to the other. I'll show you how it looks like in a moment. And in this case, we can also see this if we plot bifurcation diagrams. So if I plot this again, we can see how the biostability region the pattern region shifts to the right, so that the pattern is transformed from the classical one to the inverted one. And we can see how this looks like if I try to simulate it in a crypt geometry, very roughly crypt geometry. So what I'm plotting here, imagine crypt is like a cylinder, right? So I just cut it in half and I open up. So there is a periodic boundary condition on those vertical sides. And I also apply a Wnt gradient, which is similar to what can be found in the crypt. And if I run the simulation for the simple case of this cell monolayer, we can already see this transition from one phenotype to the other. So in the top part, we would have the classical pattern, what we call, and in the bottom, we have the pattern which corresponds to the crypt. So we could actually get it. And since I was advertising you this type of talk as something that is good to incorporate in the agent-based simulations that many people do in mass biology, in this case, if we have the subcellular signaling going on, so we know which cell corresponds to which phenotype, we can easily incorporate different phenotype-specific behaviors. So in the case of the crypt, what is known is that the Punnett cells, which are green guys in here, you remember they were very big. So they have a bigger volume, or if you look at the projection, this would be like larger area. And the stem cells, they're much smaller. So you can easily incorporate it. I didn't go much, like I didn't go really far in here because in reality the ratio is even bigger in this case. But here already the cells which are green, which acquire the Punnett cell phenotype, they in reality they have a larger area compared to the stem cells. And then you can add, of course, more and more different phenotypic behaviors, and you can incorporate it in different models that you use. So, and that's it, just to give you some summary and the outlook. So depending on which specific biological context it is found, notch signaling can generate different patterns. And I even mentioned that, the pattern can be of a stripe type, although I didn't cover it in this talk. Then the patterns that are obtained, they don't have to be limited to 2 phenotypes, although I was mentioning only 2 phenotypes. For example, there is modeling work related to epithelial to mesenchymal transition. This is the when epithelial cells acquire some motile phenotype, which is rather invasive. So in this case, there is some hybrid phenotype that has been identified experimentally and also in a modeling work. So in this case, there would be 3 phenotypes. And exactly, as I said to you, that it is good to use those cell signaling models instead of just determining rules for different cell types that you introduce in your agent-based simulations. So because they can more accurately represent the function of cell, because it's exactly perceived from the cell environment and not just determined by what the modeling researcher is telling the cell to do. And finally, if you think that those type of cell signaling networks are too complex to be included in these agent-based simulations, in reality, there are different efficient coarse graining methods. So you do not have to sacrifice the description of the system, but you can reduce its numerical complexity. And we've also been working on this. So if you're interested, I can let you know about those. And just to mention people who have been working on this with me, Tomás, who is from the CRM, and Helen and Philip from the University of Oxford. Thank you.